Okay, we are going to continue on with chapter 2, and I'm going to start throwing a lot more information your way, guys. So feel free to break these up and watch these in littler snippets, or pause these wherever you need to look up terms or Google anything that might not be clear enough. Okay? So we've talked about basic chemistry, we've talked about chemical bonds, we've talked about what it takes to build bonds, break bonds apart, those sort of things. So now we are going to get on and talk about the chemicals that we readily use in our body. The first one of those is going to be sugars. We're going to talk about all kinds of different macromolecules used in our body. So these are organic chemistry drawings of this compound. Okay, so glucose is written in organic chemistry is this way: fructose, galactose, deoxyribose, and ribose. Okay, so these are 5 really important monosaccharides in the human body and in the mammalian system in general. Okay. We have 4 other different types of macromolecules, so large molecules that are stuck together, that we're going to talk about. Okay, the first one is carbohydrates, which those are some of the saccharides that we talked about. We also have lipids, which are fats, proteins, which are amino acids, and nucleic acids, which those are used to make up our DNA. Okay. So, carbohydrates. Carbohydrates is a type of macromolecule, large molecule, in which monomers, or individual units of sugars, are put together. Okay, so you have monomers of sugars stuck together. And we have 3 major groups based on their size. We have, and those are glucose, fructose, and galactose. Okay, so those are the 3 different types of monosaccharides. The smallest kind of sugar is the single unit. These monosaccharides are the building blocks of more complex sugars. So all the sugars in the world that we're going to ingest are a combination of these 3 basic: glucose, fructose, and galactose. Glucose is your syrups and your honey. Fructose are things that are found in fruit. And galactose is found mostly in dairy products. Okay. We have monomers of sugars that are going to be joined together to make disaccharides. Dyes, too. Disaccharides are double sugars. They are formed by linking the 2 monosaccharides together. That's done by a dehydration synthesis. So remember, that's removing water from this reaction. Okay, so maltose is glucose and glucose together. Sucrose is glucose and fructose. And lactose is glucose and galactose. Remember, galactose was found in dairy, so that's where lactose comes from. So those that are lactose intolerant, you can think the galactose. Okay, here is a reaction of how to form a disaccharide. Now, I'm not going to ask you via an image, identify this molecule. I'm not going to ask you to recreate dehydration synthesis of sucrose, but you guys need to be aware that sucrose is made up of glucose and fructose, and it's created through a dehydration synthesis. You also need to be aware that sucrose can be broken down through a hydration reaction into glucose and fructose. Okay. I probably wouldn't go as far even as putting this on here and asking you what kind of reaction this is. That's a little much, but I just, there's a lot of information out there. I want you guys to start being familiar with how it's represented in the world. Okay, we've got 3 important disaccharides. Remember, di is 2, so it's 2 sugars put together. Those 3 are sucrose, lactose, and maltose. Okay, sucrose is glucose and fructose. Lactose, we talked about, is the galactose in glucose. Maltose is 2-glucose. Okay, polysaccharides. These are what we encounter in our life, mostly when we're talking about nutrition. Okay? Polysaccharides are long chains of sugar monomers, mono meaning one, a single sugar linked together through dehydration synthesis. Okay, so you pull out a lot of water in order to make polysaccharides. Okay, chains can be as few as 100 or up to thousands of sugars linked together, depending on the complexity of the sugar we're talking about. Okay, so here are some polysaccharides. One of the more simple ones are amylose, and some of the more complex are starches, glycogens, and cellulose. Okay, plants have their rigid framework of their cells made up of cellulose, and these are very complex polysaccharides. Starches typically are found in things like potatoes. Glycogens are typically found in things like grains. Okay? And these are all complex sugar molecules that are lots of sugars stuck together through chemical reactions. Okay? But we can ingest these and we can break apart all of these bonds. These are little sugar bonds, and we can get energy from them, and we can get ATP from them. Lipids. Okay, lipids are fats. It is a macromolecule, so a large molecule, consisting of carbon and hydrogen atoms that are linked together through non-polar covalent bonds. So we mostly talked about polar covalent bonds that have slightly positive and slightly negative charge. Non-polar covalent bonds are very happy non-charged bonds. Okay. Lipids being non-polar, they are not attracted to things like water. In fact, most lipids are very unreactive with water, making them hydrophobic or water-fearing. You guys have all done this. Take a little bit of vegetable oil, which is a lipid, put it in a glass of water. What's the oil do? It floats to the top and bubbles up and tries to stay away from water. You cannot easily get lipids to float in water because they have the different covalent bonds. The polar bonds of water do not play happy with the non-polar bonds of fats or lipids. Okay, so the 4 different types of fats that we're going to look at are triglycerides, which are the typical fat that we think of. Phospholipids, we're going to talk about phospholipids a lot because phospholipids make up our cell membranes. We have steroids. Yes, I'm talking about estrogen and testosterone, those kind of steroids. They are a type of lipid. And the last type of lipids are the eicosanoids, and we'll talk about those later. Okay, triglycerides. Triglycerides are very large lipid molecules that are made up of 2 monomers, so 2 single fat groups put together. So an example of one is bonding glycerol and fatty acids together. Okay? They are very concentrated. They pack together very tightly, and they have lots and lots of energy stored in there. So those are very good for energy storage. Triglycerides, meaning 3, are composed of a glycerol head and 3 fatty acid tails. So that's where it gets the tri name from. These are formed through a dehydration synthesis. We don't need to worry about that too much, okay? But a glycerol sticks together with these 3 fatty acids to create this triglyceride. And you're going to see this triglyceride a lot. So this is the glycerol head and the fatty acid tails. A fat molecule. Fat consists of 3 fatty acids linked together with 1 glycerol. Okay, so we have 2 different types of fatty acids. We have got saturated and unsaturated fatty acids. So again, we've got our glycerol head up here and our fatty acid tail. If all of the tails are straight due to all single bonds, it's considered a saturated fat. If there are any double bonds along this fatty acid chain, it causes it to be kinked. That kink prevents them to pack really close together, and that's going to cause a reaction, a chain reaction we're going to talk about here in a second, okay? So fatty acids with the double bond, so this is our glycerol head and our 3 fatty acid tails that make up the triglyceride. You can have one or all 3 of these having this double bond kink. That double bond kink is going to prevent them from stacking really close together, and it's going to prevent them from becoming a solid at room temperature, okay? So, unsaturated fats are fats with double bonds, so that's that kink, okay? Saturated fats have that kink, okay? Unsaturated fats have the kink in the tail. Saturated fats have no double bonds, so they can pack together as close as they possibly can. So think saturated meaning fully consumed. Okay? Saturated fats having no double bonds, they can become solid at room temperature. So lots of our animal fats are saturated fats. They have no double bonds, so they're solid at room temperature. That's like bacon grease, lard, butter, those kind of things. So anything that is solid or relatively solid at room temperature, coconut oil is one of them too, it's considered a saturated fat. Anything that is liquid at room temperature is unsaturated or monosaturated, okay? So it has at least one double bond. Those are things like peanut oil and olive oil. They have at least one double bond causing a kink, making them liquid at room temperature. Polyunsaturated, meaning many unsaturated bonds, so many of the double bonds. That's things like canola, corn, and soybean oil. They are all liquid at room temperature. Okay, let's go on to talk about our phospholipids. Phospholipids, like I said, we're going to talk about these a lot this year, so pay attention, please. They make up the cell membrane. They are, phospholipids contain both polar and nonpolar parts. So this results to having water-loving and water-hating or water-fearing parts. So here's our phospholipid. Okay, so we have the head of the phospholipid, which is going to be polar, and that's going to be hydrophilic, which is water-loving. So the head of the phospholipid is water-loving. The tail is these fatty chains. Okay, so the fatty chains are nonpolar, meaning that they are hydrophobic and they fear water. So you have a water-loving head and a water-fearing tail. We're going to talk about these a lot more. These phospholipids will group up, and we'll have the hydrophobic tails joining together, and the hydrophobic heads bond, sort of, not really bonding, but kind of bonding with the water in our environment, and that's what creates our plasma membrane. Okay? Steroids. Steroids are rings of 4 carbon atoms joined together. We can synthesize these steroids from cholesterol, which is a type of fat, and we can break things down and put things together to make our sex hormones and cholesterols and all kinds of other steroids. Okay, the last one we're going to talk about is an example of our lipid-derived prostaglandins. Okay, these are all going to be an example of how we modify responses to the hormones, the inflammatory response, regulate our body temperature, and those sort of things. Okay, those were all fats. Let's go on and talk about proteins. So we've covered sugars, we've covered fats. We're going to talk about proteins next. Proteins are going to contain our carbon, hydrogen, oxygen, and nitrogen. The monomers, or the single units of the proteins, are amino acids. We have 20 different amino acids in our body. Okay, I am not going to ask you to memorize all 20 different amino acids. That's just ridiculous. Okay, but you need to understand that a protein is made up of monomers of amino acids. Okay, these amino acids can link together in many, many different orders to make all kinds of different proteins. That's the big thing you need to know. Okay, so this is an amino group. So this is an amino acid. Our amino acid is made up of an amino group and a carboxyl group. Okay, that structure is really important. We're going to go on. That's where that chemistry lesson comes in handy. Okay. Okay, so we've got our central carbon, and that's going to connect to the side chain. We've got our amino group and our carboxyl group. And all of these are going to vary in different results of all of our different amino acids. So it's the R group, this side group, that's going to make up the different structures of the amino acids. Okay? So we combined different amino acids together through a dehydration synthesis. So we got to remove water in order to make a peptide bond. Okay? So a bond between two amino acids is a peptide bond. We have different types of proteins. We have dipeptides. Di is two. Dipeptides form from two amino acids joined by a covalent bond. And polypeptides are chains of amino acids. So when there's just two of them, it's a dipeptide. When there's many amino acids, it's a polypeptide. Most we're going to talk about are polypeptides. Okay, just a different view of the peptide bonds. Okay, we have levels of proteins. Proteins are very complex structure and molecule in our body, and it's extremely important to understand. Okay, so proteins are a string of amino acids that then are going to curl up to create a mass, depending on their strength. Okay, so when we look at a protein, we're going to break it down into its different levels of organization, just for complexity and ease of understanding. So the primary structure of a protein is when we're looking at all of the amino acids flat on the table, straight in a straight line. Okay, so we just have a list of our different amino acids all put together. Okay, from there, our proteins are going to start to group up. Okay, so the amino acids are then going to start twisting or folding together, and this is going to create a secondary structure that's either going to be an alpha helix, which helix is circular, kind of think of the DNA, right? Okay, an alpha helix, or a pleated sheet. So if you take a piece of paper and just start folding it back and forth, that's going to be a pleated sheet. Okay, our tertiary structure is a three-dimensional shape of these protein, either sheets or helices, put together to create a globular structure. Okay. And then a quaternary structure is when we have multiple polypeptide chains put together. Okay, so I'm going to go back and go over this again. So we have our amino acids. Our amino acids group together to make a chain. That's the primary structure. That chain is either going to curl up into a spiral, or it's going to form up into a pleated sheet. Okay, let's go on with the spiral. The spiral is then going to organizingly loop itself together into this three-dimensional tertiary structure. Okay, once you have that tertiary structure of that protein, it's going to bind together with another polypeptide chain in order to make a quaternary structure. Now, not all polypeptide chains are going to make this quaternary structure, but all quaternary structures are going to be made up of two or more polypeptide chains. Okay, confusing, I know, just roll with it. Okay, so here's just a different way to look at it. We've got our amino acid chains. They're going to loop up into our spiral. That spiral is going to fold back onto itself to make its tertiary unit. and then those tertiary units are going to glob together with others to make a quaternary. Okay, so this example is of hemoglobin in our blood. So the protein is going to be transcribed from our DNA. It's going to be read in our bone marrow. That bone marrow is going to say, hey, we need more blood. Okay, so it's going to kick out the amino acid chain to make hemoglobin. Okay, so it's going to kick it out, and as it kicks it out, it's going to fold up properly into the secondary structure. That secondary structure is going to then loop itself together around the heme units, and then those heme units are going to bond together to make this whole hemoglobin subunit. Okay, we'll get there, just roll with it. Okay, protein denaturation. So we talked about how to build proteins. Now we're going to talk about how to take them apart. Okay, so the function of a protein depends on its ability to recognize and bind to some other molecule. Some hostile environments, such as heat, acids, or salts, will change the 3D shape and destroy its ability to function. So when we denature a protein, we are going to make it fall out of this tertiary and quaternary shape. Okay, so when it can no longer be in its pretty designed shape, it's called denaturation. Okay, enzymes. I told you we were going to talk about enzymes a lot. Enzymes are proteins. Okay, so an enzyme is a whole bunch of amino acids in a particular shape that is going to act as this little canister that's going to catalyze reactions. Okay, so we've got two parts of the enzyme. Don't worry about this too much. I want you to know what they do, not what they are more. Okay, so they've got a protein portion and a non-protein portion. The protein portion is all those amino acids. Then it might be surrounded by something like iron, zinc, magnesium, calcium, and that's going to aid in that chemical reaction. Okay. Enzymes are there in order to reduce the activation energy. Okay. So they are highly specific. So that canister is only going to open for specific substrates. Okay. So it's only going to open for specific things. It is going to only allow the one to enter, and it's going to speed up only that one reaction. So we have enzymes that function very specifically. Okay, they're also very efficient. They can speed up the reactions 10 billion times faster and easier than if we were just set them on the lab table. Okay, the enzymes are also under cellular control. So the rate of the synthesis of the enzyme or that protein is under the control of the cell's gene. So if we don't need to do that chemical reaction, our body will not make that protein for that enzyme, for that reaction to happen. Okay, so it's the genetic material that's going to determine how much of the enzyme is available. Okay, substances within the cell may either enhance or inhibit the activity of the enzyme. So we have mechanisms. Sometimes we need that enzyme pre-built because the reaction might have to happen really quickly, more quickly than it takes to make a protein. But we can have things in the cellular environment to hold that enzyme in a specific shape, in reactive shape, until it's needed. Okay, and so that activation or inactivation could be determined by something in the chemical environment of the cell. Okay, so it needs a specific key in order to allow that enzyme to function. And we're going to talk about this a lot, so just roll with it. Okay, so remember, enzymes are proteins. Okay, so this protein glob. Okay, in this example, the enzyme is going to collect two different substrates. So remember, that's the beginning part of our chemical reaction. Those little substrates are going to float around and they're going to lock into. Okay, so those substrates are going to bind to the active site on the enzyme. So it's kind of like the little kid toys with squares and circles on top that go into the box. So it takes the right sized amino acid or substrate in order to put into this activation site. Once it's in the enzyme substrate complex, these terms are important to know, okay? Then this enzyme is going to kind of forcefully shove the chemical reaction together and to happen, okay? Remember that these are all dehydration reactions, and so it releases water, and then that amino acid or that reaction is going to have been formed, and you get our product. In this case, it's a dipeptide, and then that enzyme is then now open in order to catalyze another reaction. So really important, we want to know the substrate. That's the pieces of the chemical reaction. Our enzyme, that's the protein that is going to aid in the chemical reaction. Once those are all together, we have an enzyme substrate complex. Okay? That enzyme substrate complex is going to take the substrates and shove them together and make the reaction happen faster and easier. It's then going to release our product, and then that enzyme at the end is going to start over and be empty again. Okay, I can almost guarantee test questions on this. Okay, so here's just another way to look at it. There's lots of different ways to draw these. So you've got an enzyme. In this case, it's got two different substrates. So let's pretend we're making table salt. So this is sodium, this is chloride. They both are going to lock in here into their different key locations. The enzyme's going to then shove the sodium chloride together, and it's going to, once it makes the table salt, it's going to kick out the table salt, and then it creates the empty enzyme again. Now, keep in mind that I just made that up, and we don't really have enzymes to catalyze table salt reactions, but I'm just trying to simplify it for you guys. Okay, the last one, we're almost done with this chapter, guys, bear with me, is nucleic acids. Nucleic acids are polymers, so many things that stick together that serve as a blueprint for proteins. So something has to, in our cells, create all of these proteins that are incredibly important. In our body, we have nucleic acid polymers that create either DNA or RNA. DNA stands for deoxyribonucleic acid. Big mouthful. I am totally okay with you guys just using the term DNA. You don't need to be able to write or spell deoxyribonucleic acid, but you guys do need to know what it is and how to recognize it. Okay. The other one is ribonucleic acid, or RNA. Okay, so DNA is the encyclopedia of our cell, the blueprints for life. Okay, DNA is going to be read in the nucleus, and we'll get there. You'll understand all this eventually. Okay, but DNA is going to be read in the nucleus, and little pieces of that encyclopedia are going to be encoded into RNA. This RNA is going to leave the nucleus, float into the cell, and that RNA is going to be read and turned into amino acids that will make up proteins. Okay, we'll get there. So, nucleic acid monomers, so individual pieces of a nucleotide, of a nucleic acid, are the nucleotides. They are made up of 3 parts. The first part is a 5-carbon sugar. In the case of DNA, that's the deoxyribose. In the case of the RNA, it's a ribose. And then it's made up of a phosphate group and a nitrogenous base. Okay, so we have our sugar group, whether it's deoxyribose or ribose. We have our phosphate group, and then we have our nitrogenous base. Okay, and these are all going to string together to make DNA or RNA. In DNA, the nitrogenous base is either an adenine, a thymine, a cytosine, or a guanine. So it's either an A, T, C, or G. We'll come back to this a lot. Okay, so in DNA, it's either an A, T, C, or G. In RNA, it's either an adenine, a uracil, a cytosine, or a guanine. So how you can tell the difference between DNA and RNA is whether it's a thymine or a uracil. Okay? So in DNA, it is going to curl together to resemble a spiral ladder. We call this a double helix. So we have two strands of alternating phosphate groups and deoxyribose sugars going upright from the ladder, okay? So you've got your phosphate groups and your deoxyribonucleic acids or deoxyribose sugars on the solid part of the ladder, and the rungs are made up of the nitrogenous bases. And that nitrogenous base is either the A, T, C, or G, because we're talking DNA. Okay? These bases are going to pair up with the opposite strand of the DNA. Adenine always pairs with thymine, and cytosine always pairs with guanine. So A always goes with T, and C always goes with G. So G goes with C, and T goes with A. Okay, here's just another look at the nucleotides. They are the building block of the RNA and the DNA. So we've got our A, T, C, G, and then the uracil and RNA. And here's just another way of looking at the double helix. So we've got our double strands connected together with the hydrogen bond. So it's a really weak bond because the DNA often has to be pulled apart in order to be read. Okay, now we're going to move on and talk about ATP. ATP, or adenosine triphosphate, adenosine 3-phosphates, is really, really important in the human body because it is the power of the cellular activities. It's the energy source of the cells. So ATP consists of 3 phosphate groups attached to an adenine and a ribose. Okay, so a sugar and an adenine. The ATP we'll talk about later and more throughout the term often goes between ATP, adenosine triphosphate, having all 3 phosphates, to ADP, adenosine diphosphate. It's very rare that we see adenosine monophosphate, but it happens. Okay, so what's really cool about this and what makes ATP special is that there's a lot of energy stored in these phosphate bonds. It's really easy to then release this energy and use the currency from this ATP, but it does take some energy to put it back together. Okay, here's just another way to look at the ATP. So we've got our adenosine portion here and our triphosphates with the different bonds. So we can break a bond here and make it ADP, and then we could break it apart again here, but we typically don't see that. Okay, formation and use of ATP, so adenosine triphosphate. It goes through hydrolysis, so hydrogen water is used in order to release the energy, okay? So we use water in order to break that phosphate bond and take it down to ADP, a loose phosphate group, and energy. It's this energy that we're really after. Okay, after we get the energy, we then have to put it back together, and it's you get much more energy out of the ADP than it takes to put back in. Okay, so you add the ADP and that phosphate group plus a little bit of energy to yield this ATP and water. So the byproduct of this reaction is water. Okay, so that's all we have for chapter 2. If you guys have any questions, please use the discussion board. There's also great resources on Google, on YouTube. Your book, of course, has lots of great videos and activities to go through. And then, of course, you feel free to reach out to me if you have any questions. Thanks, guys.